Okay. So then, first of all, are there any questions about chapter 20 lecture or chapter 19? No? Okay. And I just want to remind you that homework 21 is due by midnight tonight. That's really on all the way back to chapter, I forget.
18.
18, I think, yeah?
Yeah.
Yeah.
Are you going to change it on La Lima? Because it still says the 13th.
It does?
Yeah.
Oh, okay, I'll note, just check that, okay? Oh, you know why it says that? I think it's because it's due on the 6th, but it's not closed because I'm letting people turn in late homework.
Oh.
But I'll recheck that again. Yeah.
Okay.
Okay, so I'm going to share the screen for thermodynamics. Okay. And unfortunately, when I do that, I cannot see what you guys are typing in the chat. The chat disappears when I do that. Oh well. Technology. So thermodynamics simply means, thermo means energy or heat, and then dynamics, how it changes around and does everything. And of course, obeys the law of conservation of energy. So the first thing we'll look at in this chapter is what we really did at the end of the last chapter. Now we're going to be a little bit more formal about it. So say, for example, we look at this reaction here. Here's essentially how this works. If I take a container and I fill it with nitrogen and hydrogen, and then I let it go to the point of equilibrium, some of this gets converted into ammonia. So at some point in time, the concentrations don't change anymore. It's at equilibrium. So I have a certain amount of each of these three entities. But if I took another container and I put ammonia in there, it's going to go in the reverse reaction until you start making a bunch of this and a bunch of this. And you know what? The concentrations of those at equilibrium would be the same. It doesn't matter whether you start with ammonia or whether you start with nitrogen and hydrogen. That's what we mean by this, okay? And so Le Chatelier was a guy who figured out how to manipulate chemical reactions that were at equilibrium. Because say, for example, your goal was to produce as much ammonia as you can, because that's an important component of, say, fertilizer. You would start with these gases, and then you end up with this, and then you can't go any further. So he was thinking, how do we get more? And so this is the way I do it, which I might mention is also the best way to do it, because I do it that way. And I'm the best. So you're reading the problem. So what happens to the ammonia concentration when the nitrogen concentration is increased? So you have to identify what the stress is. And the stress is that the nitrogen concentration was increased. So I put the stress on the bottom. Okay? So in other words, it was just sitting there at equilibrium. You added more reactant, so something's going to happen to get to a new point of equilibrium. So the result will be this arrow is either going to go towards products or go towards reactants. There's no other possibility. But in just terms of logic, if you add more nitrogen, when you're adding more reactant, you would expect to get more product. So now I've decided that that arrow is going to go in that direction. And once I set the direction of that arrow, I can figure out what's going to happen. As I add more nitrogen, I'm going to shift the reaction this way. All things over here are going to go up. That means I'm going to make more ammonia. The temperature is going to go up. And why do I have this decreasing over here? Well, that's because of stoichiometry. Every time one nitrogen goes this way, it takes 3 hydrogens with it. So this concentration is going to go down. Okay. So hopefully that makes sense. Now we learned that equilibrium constants are constants. So that means here's the equilibrium expression, products over reactants. Notice that the 2 is there because there's a 2 there. There's a 3 there because there, right? So what this really means is that 2 molecules of ammonia have to collide in order to go that way. So I'm just combining like terms, actually. But the equilibrium constant is going to be a constant. What happens if I increase the nitrogen? So numerically now, if I increase this, something has to change with these other concentrations because that's still going to be a constant. So one way to alleviate the constant from changing by adding more of this is numerically, if I increase the denominator, I increase the numerator. And does that make sense? Yes, it does. Also numerically, if I were to increase this, I could counteract that by decreasing this. And does that make sense? Yes, every time one of these goes this way, it takes 3 hydrogens with it. So in other words, Le Chatelier's principle, you can look at it in terms of what's called the mass action shift, what happens, and also by looking at it, what happens to keep the constant a real constant. So look at the next problem. What happens to the ammonia concentration when the nitrogen is decreased? So this time I'm reducing the nitrogen, that's the stress. And so I'm going to activate this reverse reaction. So whatever stress you do, it will shift in order to try to undo what you just did to it, because it wants to get right back to equilibrium. So that means that the arrow is going to shift that direction. So everything over here is going to go down, and everything over here is going to go up, except you're reducing that as you make it happen. Okay. And what happens to the ammonia when the temperature is decreased? So the stress is that you're reducing the temperature. So what's going to happen? It's going to shift in that direction to try to replace that heat energy so it can stay at equilibrium. So once I establish that arrow, then everything over here goes up, everything over here goes down. Okay. This one is a little bit more esoteric. What happens to the ammonia concentration when the pressure is increased? So the stress is that the pressure is increased. Now, we already stated that these were all gases, right? So if you increase the pressure, it's going to try to counteract that by decreasing the pressure. So what do we know about PV equals nRT? Well, if you have R is a constant, T is a constant, and the volume of the container being a constant, that means that the pressure is proportional to the number of moles. So over here, I have 4 moles, and over here, I have only 2. So how do I counteract this increase in pressure? I can counteract it by reducing the number of moles of gas. So the reaction will shift in that direction because I'm going from 4 moles to only 2 moles. So we looked at this one reaction in kind of a lot of detail. And then I'm going to take that same reaction and we're going to talk about what's called the Haber process. It was done in 1908 using Le Chatelier's principle. And what the Haber process does is it allows us to make huge amounts of ammonia. So why is this important? Ammonia is used in manufacturing. The most important use of ammonia is just a way to get nitrogen into plants. It's a fertilizer. Okay. So one of the most important fertilizers is ammonium nitrate because it gets both 2 chemical forms of nitrogen into the soil. So say you're hired as a chemical engineer. Now, whenever you put the word engineer onto your title, you're going to make more money. Sorry, my finger muted me. So remember, I went to my chancellor one time and I asked him if I could change my title to professor engineer and then get more money, and he didn't think that was such a good idea. So you get hired as a chemical engineer. Your job is to make more money for the company. If the company is involved in producing ammonia, that's your job. Your job is to make this arrow go in that direction so you can maximize the production of ammonia. So knowing that, what would you do with the partial pressure of, say, hydrogen? Well, you'd increase it, right? But you also have to be careful because if you ever had a breach in the container, then nitrogen would react with oxygen. Now you have an explosive situation. And if you're the chemical engineer and you blow up the chemical plant, then you're probably not going to get that raise you were hoping for. What about nitrogen? Well, yes, you increase the pressure of nitrogen too, wouldn't you? So now you've done everything that there is to do with this. But notice, as I'm pushing the reaction this way, what's happening to the temperature? The temperature starts to go up. In a high temperature, you're activating the reverse reaction, so you don't want to do that. So here we have an exothermic reaction. It's going to keep getting hotter and hotter and hotter. So what you need to do is remove that heat energy. You need to cool it down. So far, we've got high pressure and we're reducing the temperature. We're refrigerating this chemical reaction. And there's one more thing that we can do, and that's to remove the ammonia. As the ammonia is formed, we remove it. So here's what we got so far. I got nitrogen and hydrogen and some ammonia gas. What I'm saying, if we lower the temperature enough, we will start to liquefy the ammonia. We won't liquefy the hydrogen or the nitrogen, we'll liquefy the ammonia. Why? Again, it goes back to attractive forces. Nonpolar molecule, nonpolar molecule. These reactants right here, their strongest possible attractive force between anything is simply London's. And what about ammonia? One molecule would have an NH, the other molecule would have an N. That's hydrogen bonding right there. So the hydrogen bonding is going to help this condense into a liquid. So now I can drain that ammonia off, put it into another container, maintain the pressure, maintain the temperature, and keep it as a liquid. Later on, I could acidify it and turn it into, say, the ammonium ion, which will now be soluble in water and won't ever run away as a gas. So there's one more thing that we can do, but let's look at the difference in equilibrium constant when ammonia is a gas and when ammonia is a liquid. Products over reactants, right? Look up when ammonia is a liquid. Why did I leave out ammonia? Because now it's a pure liquid. We omit it from the equilibrium constant. Why do we do that? Guess what? Equilibrium constant is now different. So we successfully use Le Chatelier's principle to overcome the limitations of the equilibrium. And here's another thing. We haven't studied this yet, but this is the rates. Here's the rate equation. So the rate of going in the forward direction is equal to some rate constant times the concentration of the 3 hydrogens that have to bang into each other and the 1 nitrogen. So when I combine like terms, the rate in the forward direction is equal to some constant times the concentration of the reactants. If I look at the rate in the reverse direction, it's going to have a different rate constant, and it's going to be dependent upon the concentration of the ammonia squared. Okay. Well, what about when it's liquefied? Notice that the rate in the forward direction is the same. I didn't affect that. But what about the rate in reverse direction? Well, there is no product because every time I form it, I remove it. So the rate in the forward direction now becomes 0 because there is no ammonia. So this allows us to make huge amounts of ammonia where we couldn't do that before. And by the way, this is probably what caused or how humans were able to avoid starving to death. There are several predictions of mass starvation because not enough food, too many people moving into cities and stuff like that, not growing food anymore. It's actually the fertilizer was the first agricultural revolution. Anyway, we are not at equilibrium anymore. We go back to this equation right here, and that reverse reaction is not in effect anymore. So now it's racing towards equilibrium, and it's not able to get there because we keep removing the products. Now, addition of a catalyst will increase the rate of ammonia production. So here's what catalysts do. So here's an energy diagram. Here's the potential energy in the reactants. Here's the potential energy in the products. So this difference right here, which is 22 kilocalories, makes it our exothermic reaction, right? So we don't really have time to go into it, but in a higher level chemistry course, we'd spend a lot of time on it. This difference right here, which we could call delta G or delta E or whatever, if we're talking about delta G, that's equal to the negative log of the equilibrium constant. You don't need to know that exact equation, but what it means is that the difference in energy between reactants and products is what determines the equilibrium constant. So, That's the reason why it's a constant, because it's dependent upon what is the reactants and what is the products. What about this? So here's the transition state. So we talked about this before. In the reactants, every single atom is meeting the octet rule and the duet rule. There's electronic stability here. There's electronic stability in the products. But in the transition from going to reactants into the products, there's not. So this is the highest energy possible when going from reactants into products. What does a catalyst do to make a reaction go faster? It lowers the energy of that transition state. It changes how the reaction happens. For example, if the catalyst is an enzyme, the enzyme will bind to one or more of the reactants, do something to them that makes them lower energy and more stable, and then let them go on to the next step instead of just leaving it to fate. So this part right here is what determines the rate. It's the energy in the transition state, the energy of the activation energy. So what does the catalyst do? It cannot change the equilibrium constant, but it will change the energy of activation, making the reaction go faster. So here's a concept. Looking at this original reaction, I put a catalyst in there. What does the catalyst do? It makes the reaction go faster to the point of equilibrium. Because I make the forward rate go faster and I make the reverse rate go faster. Looking at the energy diagram, by lowering this energy right here, it's easier to get to products and it's easier to get back to reactants. So if I add a catalyst to a reaction that is already at equilibrium, nothing happens. So here I reverse this. So I reverse the reaction a little bit. So what happens to the concentrations of gases when the temperature is increased? So that's the stress. I increase the temperature. That's one of the reactants. I'm going to push the reaction that way. Once I decide the direction of that arrow, everything else just follows along. So everything over here will go up. This will go down because we're moving everything in that direction. What happens to the concentration when the pressure is decreased? So if you decrease the pressure, then it wants to increase that pressure to get back to where it was at equilibrium. So how do I increase the pressure? Well, here I have 2 molecules of gas. If I go this way, I got 4. So if I go this way, I'll reduce the pressure. Here's one way that photosensitive lenses work. So in the glass is embedded, or plastic is embedded, silver chloride. So right now, there's not enough of it to block any significant amount of light. But when light comes into the picture, it's part of this equilibrium. Light is a reactant, pushes the reaction this way. So what we do is we form elemental silver, which blocks the light. We form chlorine gas, which is now trapped in the plastic or in the glass. If you take away the light, then it goes back this way. So anyway, what about older glasses? So the glasses that I have now, they're starting to stay darker all the time. And when I look back at the pictures that I took when my kids were little, and this technology wasn't as good, and you can't just go buy a new pair of glasses whenever you want. It's kind of embarrassing because all the pictures of me and my kids, I have dark glasses on. They didn't revert to clear anymore. So what happens is you start to lose the chlorine. The chlorine gas seeps out of the plastic, out of the glass. So as you remove that chlorine, you're shifting everything this direction. So it just starts to stay darker. Okay, so let's see. See if there's anybody doing chat. Okay, so nobody has any questions? Okay. Back to screen sharing. So collision theory and the transition state, very, very simple concepts. So here I have a chemical reaction. You can see it's an endothermic reaction. I have to add heat energy to get from reactants into the products, right? So that's this value right here. Don't worry about the positive or negative right now. But what I want to say about collision theory is actually very simple. Here's two molecules that have to collide in the exact right orientation. And I don't have anything with me right this second. Usually I find something in the lab to grab and use as an example, but don't have anything simple right here. So these two molecules have to collide in exactly the correct orientation. And they have to collide hard enough to get into what's called the transition state. So in the transition state, the bonds that are to be broken, which are these right here, are halfway broken. The bonds to be formed, which are these two right there, are halfway formed. So why is this at the highest energy right here? Because you're not meeting the octet and the duet rule. You don't have electronic stability. So if they bang into each other, but not enough energy to come up over here, they simply revert back into reactants again. If they collide with enough energy and in the right orientation, they'll get over here and there's a 50-50 chance of going one way or the other. If they go this way, then you finish breaking these bonds and forming these new bonds. And now you have a product that has more potential energy than the reactants do. So again, just to reiterate, the equilibrium constant for this reaction depends upon the delta G. Okay, so here's another way of writing it. I know I just realized I wrote it two different ways. Both of them are correct. So negative RT times the natural log of equilibrium constant, or just negative log to the base 10 of the equilibrium constant. Oh, okay. Here it is. So the collision has to happen with the correct orientation to be fruitful. Has to be hard enough to get reactants into the transition state. And then this is, of course, the energy of activation. And that is what determines the rate of a chemical reaction. So what does a catalyst do? A catalyst does not affect energy of reactants or products. It doesn't affect equilibrium constant. What a catalyst does is it interacts with the reactants in such a way as to lower the energy of activation, to lower this bump right here. That's what it does. So I tried to give you an example of an enzyme. So what can an enzyme do? Well, it's going to lower the energy of activation, just like any other catalyst will do. And in the Biochem 341, which I'll be teaching this summer, or the 244, which I might teach spring. I guess we teach it spring at KCC. Much more detail, a whole chapter on it. So what does the enzyme do? Well, it concentrates the reactants on the catalyst. So what does that mean? Here's the active site of the catalyst, the active site of the enzyme. After billions of years of evolution, the design of this protein is such that it forms a strong attraction between itself and the reactants. And when I say attractive forces, right, I'm talking London's, dipolar, hydrogen bonds, and ion to dipole, right? And so why does it get better and better and better? Well, because say you're a lizard living in your backyard. I'm not saying you are a lizard, but that's a good example. What if another lizard subspecies comes in, it's in competition for food and everything else with this other existing lizard, and it has an enzyme that binds its reactants stronger and makes the reaction go faster? That could very well give an advantage to that new subspecies of lizard. If it does give an advantage, either catching prey or a mate or whatever, then its genes are more likely to be passed on to all the other lizards in your backyard as time goes on. That's called evolution. If, for example, there is a mutation that made that chemical reaction go more slowly, less efficiently, well, then you would lose out to some other lizard with a better metabolic pathway. So that means if there's two reactants, say for example A and B, A can bind to the enzyme, B will bind to the enzyme, and now they're concentrated. They're right next to each other. It brings them into close proximity to each other. And so it puts the reactants into the correct orientation on the surface of that enzyme. Now, every collision is going to be fruitful. There is no collision that's not in the right orientation. And then it puts strain on the bonds to be broken. So what does that mean? If you take the next semester, then you'll see that proteins are large. They can go through what's called conformational changes, which means that they change shape. So here's what that means. Say I'm the enzyme. This is my reactant. We call them substrates. It binds to me. Why does it do that? Because of the design of the enzyme. Now, when it binds to me, there's conformational changes, which means single rotates, bonds about, excuse me, rotation about single bonds. Here I can grab this. Here's a conformational change. See this? I'm only doing it with this one single bond right there. That's the conformational change. So say it's like this, it bonds to me, it goes through a conformational change. I also go through a conformational change. And that movement puts strain on the bonds to be broken. So these first three are in all the biochemistry textbook. The fourth one I added because I don't know why they don't add this. The enzyme also supplies chemical functional groups. Say the chemical reaction that the enzyme works on is a redox reaction. It transfers electrons from one item to another. I will almost guarantee you that that enzyme will have a way to take electrons from one reactant, hold on to it, and then later on give it those electrons to the other reactant. What if you have an enzyme reaction that's catalyzed by an acidic environment? And there's many of them in biology. The pH at the active site of the enzyme can be way different than the pH in the solution. All I have to do is take a bunch of acidic functional groups from acidic amino acids and load up this area right here. So outside might be 7.35 physiological pH, but on the surface of this protein, it could easily be 1.5 or 2. So here's an example of a methyltransferase. And so there's lots of methyltransferases. One of the most important ones now is the methyltransferase that's used to methylate sections of DNA to turn on and turn off genes. But they all work in a similar manner. If you were to look at all the different amino acids, you would see that the types of chemistry that's possible is actually very limited. So what nature has done in order to expand the chemical capabilities is to use what's called coenzymes or cofactors. So these are the vitamins and the minerals. So if I'm an enzyme that catalyzes a redox reaction, I'm going to have something in there like an iron. So molecule A comes to me. I take its electrons. My iron goes from a +3 to a +2. That bounces away. Now B comes in, and my iron atom gives electrons to that, causing it to be reduced. So I've used iron to make the chemistry possible. In this particular example, this enzyme has a unit of folic acid on it. So folic acid is a vitamin. You need it. It's involved in several metabolic pathways. Here's what it does. So here's the chemical reaction. It takes a methyl group from A and puts it onto B. So it separates it into separates it into steps. So here's enzyme with this folic acid cofactor. It binds the first reactant that has the methyl group on it. It holds on to the methyl group using the folic acid. Now, first molecule can go away. So now the enzyme is loaded up with methyl groups. The new reactant comes in, then it simply transfers the methyl group to that new reactant, and there's your final product. So here's a schematic. How does this fit into a metabolic pathway? So here's a schematic of a metabolic pathway. So reactant A is turned into B by this enzyme that works on A. B is then converted into C by this other enzyme. And C gets turned into D, which may go this way to form some final product, or maybe go this way to form another product. You're constantly synthesizing things in your body, aren't you? What would happen if this enzyme right here was not functional? It could be unfunctional for 2 reasons. One, there could be a genetic defect. The amino acid sequence is wrong. That would be a genetic disease. The only way to treat that would be to alter the genetics. The other possibility is that that enzyme B requires a cofactor that you have to get in the diet. For example, folic acid is one of those. If we don't eat enough greens, we don't eat enough salads, we will not get enough folic acid. So if you don't have enough folic acid, you will not have an active enzyme there, and now you're suffering the consequences of all that. It doesn't necessarily mean yes or no, the reaction is going to happen or not, because it just depends on if you're totally depleted of folic acid or not. If you're totally depleted of folic acid, you'd simply be dead. If you don't have enough of it, then you have a compromised metabolic pathway. And you now have a disease. It's a nutritional related disease. Okay, so let's see if there's any questions here. Any questions about any of that? You can tell I'm a biochemist, because biochemistry is the best. Okay. So now reaction rates. We'll look at it a little bit more closely about reaction rates. So if we look at this reaction right here, the rate in the forward direction is going to be equal to a rate constant for the forward direction times the concentration of A times the concentration of B. So let's look at it mathematically. What if the concentration of A was 0? Then the rate would be 0. Makes sense, right? What if I doubled the concentration of A? I have a certain rate. Now I double the concentration of A. I should double the rate, right? What about reverse reaction? So here, two molecules of C have to collide with one another. That's why it's dependent upon both of their concentrations. It just so happens that they're like terms which can be combined. So what would happen to the reverse rate if I doubled the concentration of C? Well, then I would quadruple the rate in the reverse direction. Make sense? So I might point out that, of course, this is theoretical. Theory is one thing. Actually doing it in the lab is a different one. So let's look at something more practical. Here's the destruction of hydrogen peroxide, okay? So the rate in the forward direction is going to be equal to the concentration of two molecules of hydrogen peroxide. They both have to bang into each other. So that's why I have this squared there. Okay, but right now that's just proportional. So how do I get the rate constant? Well, what I do is I measure the rate at a given concentration. Here's the concentrations. And how do I get that rate constant? It's simply the mathematical definition of that. So that's where the rate constant comes from. It has to be measured. It's really nothing more than a proportionality constant. And we always use a lowercase for rate constants because we always use an uppercase for equilibrium constants. So the actual rates depend upon exactly how the reaction takes place in a step-by-step manner. And that is referred to the reaction mechanism. So anyway, what I've found over the years in my biochemistry courses, where they're supposed to know this stuff, even the higher level, especially in the higher level ones, they can do all the Michaelis-Menten kinetic calculations. They know all this stuff how to do. Then you ask them a really simple question. What is a rate? They don't really know. But to me, it's more important to know the basics. And the basics are actually pretty simple. So here's the chemical reaction that we're going to look at, the same one right here. So I set it up. So in the container, I have only hydrogen peroxide. That's how I start the reaction. So right now, there is no oxygen. I'm right here. And as time goes on, I start to produce some water, some oxygen as products, and I still have some hydrogen peroxide left. But if we just look at the oxygen, as time goes by, I'm making more and more oxygen. So I will get through a certain period of time where there's a straight line passing through the origin. And after a certain period of time, it starts to taper off. And you see why it's getting level like that? Because it's going to equilibrium. Eventually it will go to equilibrium. But what I'm interested in is the initial rate is this straight part right here. So I take the initial rate by drawing a straight line through that first several points. And then what is the slope of that straight line? It's the change in y divided by change in x. In this case, it's the change in millimoles of oxygen produced per minute, which is the rate. So anyway, now we know what a rate is. And going back to this, the rate has to do with this transition state right here. So what kind of factors affect the equilibrium rates? If I increase the reactant concentration, I increase the number of collisions, and therefore I increase the rate. If I increase the temperature, I'll increase the rate of collisions, in other words, the collision frequency, and the energy of the collisions will be stronger. So in general, an increase of 10 degrees in temperature about doubles the reaction rate. You ever put a cockroach in the refrigerator? Well, so you open up the door and the light goes on because the light is off when the refrigerator door is closed. And if there's a roach in there, you'll see the roach tries to run away, but he's like running very slowly. But if he warms up, then he'll be running faster because it all has to do with that. Then a catalyst will lower the energy of the transition state by changing how the reaction happens. So that will also affect the reaction rate. So going back to hydrogen peroxide. Hydrogen peroxide slowly breaks down to form water and oxygen. You have bought probably a 3% solution of hydrogen peroxide in water at Long's or something like that. And you notice that it's in a completely dark bottle because light will help activate this chemical reaction. It's also in a completely plastic bottle because certain metals, especially iron, will cause this reaction to speed up dramatically. So you say you have a cut on your arm. If you put hydrogen peroxide just on your skin, it doesn't do anything. But if it touches where there's blood, then you see all this bubbling happening. It's just super active. The bubbles you're seeing are the evolution of oxygen gas. And the reason why it's going fast is because the iron that's in hemoglobin is available for hydrogen peroxide to interact with. So your blood heme becomes the catalyst to make that reaction go faster. Now, if you're an aerobic organism, you have to survive being in the presence of oxygen. Look at the reverse rate here. You are in an environment with oxygen. If you're not, you're dead. You're made up of water. If you don't have enough, you're dead. Those two inevitably will go to form hydrogen peroxide. So you are making hydrogen peroxide right now as we speak. The problem with hydrogen peroxide is that every so often it breaks down not this way, but this way. It breaks into what's called hydroxyl radicals, which we discussed before. So if you're worried about getting a bacterial infection from your desk, you're not going to get an infection from an anaerobic bacteria, because anaerobic bacteria cannot survive in the presence of oxygen. You know why? Because it's the hydroxyl radicals that kill it. It destroys the DNA. But because you're an aerobic organism, you have an enzyme called catalase. So here's the best scenario that I've been able to steal from the internet and modify. So here's catalase right here. Catalase has in it a cofactor of iron. It's an iron-dependent enzyme. So if this could be allowed to be considered to be hydrogen peroxide, that comes in, it gets sucked into that lock and key type situation right there. In other words, it's highly attracted to the enzyme because of the shape of the enzyme after millions and millions of years. So it's attracted by London's dipolar hydrogen bonds and ion to dipole attractions. It gets sucked right in there. And the perfect orientation is that the bonds to be broken over here are broken by putting strain on it and then being pulled up right up against that redox component, okay, causing it to react. So when you break it down safely into oxygen and water, then you don't produce any free radicals like the hydroxyl radical. So anybody have any questions at this point? So now that we understand rates, we can go back and take another look at the equilibrium constant. So we'll use this chemical reaction again. So the theoretical rate in the forward direction is this. The reverse rate would be that, right? So equilibrium is defined when you reach the point where the concentration is no longer changed with time. And people tend to think that, oh, well, if it's not changing, then nothing's happening. In other words, they think like it's static, but in fact, it's dynamic. The concentrations don't change because the rate in the reverse reaction becomes equal to the rate in the forward direction. And these rates are not zero. In a static situation, the rates would be zero. But these reactions never stop working. So here's a couple definitions of equilibrium. So here's time versus concentration. So if we look at the beginning, right, we have hydrogen and iodine. We don't have any hydrogen iodide yet, right? So we look at the concentration of hydrogen iodide at 0. And as the reaction proceeds, that increases to a certain point. And at that point right here, the concentrations no longer change. So this is pre-equilibrium and this is at equilibrium. If you look at the concentration of, say, one of the reactants, in the beginning, it's going to be high. But as it gets used up, then it starts to decrease. But then it tapers off and it gets to the point right here where it stays stationary with time again. So one definition for equilibrium is that concentrations do not change with time. So now, what happens with the rates? If we look at the beginning, the reverse rate is 0 because there is no product to break down. But as the reaction goes along and you're making more and more hydrogen iodine, the rate will increase because the concentration of hydrogen iodine increases until you get some point where the rate stays the same. The rate will stay the same because you have a constant amount of hydrogen iodide. What about the rate in the forward direction? It's going to be high in the beginning because that's where you have the highest concentration of reactants. And as the reactants start getting used up, the rate will decrease until you get to some point where it stays the same. So notice that at equilibrium, the rate in the forward direction is now equal to the rate in the reverse direction. And those rates are not 0. So here's the derivation. I put it in a red box to show that equilibrium constant is a constant. So the rate in the forward direction now equals the rate in the reverse direction. So I substituted with what they are, right? And all I did was I put all the constants on one side and all the variables on the other side. So I divided both sides by KR, right? Divided both sides by this. Now, if you take a constant and you divide by another constant, what do you get? Another constant. It's called the equilibrium constant. And look at the definition. It's products over reactants. So let's put this into perspective with a problem. Write the forward and reverse rate reactions in the forward and reverse equilibrium expressions for this reaction. So remember, we're almost always using homogeneous equilibrium. So the rate would be the rate constant in the forward direction times the concentration of B raised to the third power, C raised to the first power. And why did I leave out A? Because it's a pure liquid under these conditions. And the equilibrium constant would be products over reactants. I left out D because it's a solid. I left out A because it's a liquid. So notice that it's 3 molecules of this and 1 molecule of this. So 4 things have to collide, right, to go into the forward direction. And then similar for the E is the product. So what about the rate in the reverse direction? All we're really doing is redefining what is a reactant, what is a product. So the rate going into the reverse direction will be equal to reverse rate constant times the concentration of E raised to the 4th power. And then, if I'm going that direction, here's the products and here's the reactants. See that? And now, hopefully you notice this. The equilibrium constant in the forward direction is equal to the inverse of the equilibrium constant in the reverse direction. They're inverse to one another. Because all you've really done is change the definition of what is a reactant and what is a product. So now if you're a chemist and you're trying to figure out how to set up a reaction you're going to do, you can go to a group of tables, look up equilibrium constants. Do you need to have the equilibrium constant for both forward and reverse reactions? No, you don't, because they're just the mathematical inverse. Okay, I put this in a red box. I'm trying it out this semester. We'll just see how I feel about it later. So I'm not going to make you do this. But I want you to realize that everything in a chemical reaction is a direct proportion. Stoichiometry is obeyed, law of conservation of mass, law of conservation of energy. That's the reason why we can do these problems. So calculate the equilibrium constant when 2 moles of A is mixed with 1 mole of B, and at equilibrium, here's the concentration of A. Given only one concentration at equilibrium, I can figure out the concentration of the other entities. So I started off making a 2 moles per liter solution. At equilibrium, it's only 1.8 moles per liter. That means that 0.2 moles per liter went that way. So using stoichiometry, 0.2 moles per liter of A, I got to get rid of moles per liter of A, get moles per liter of C. That means that I made 0.1 moles per liter of C. Similarly, I made 0.3 moles per liter of D. So now I have the concentration of A, C, and D at equilibrium. I need to calculate this now. So it's a little bit more complicated. To use 0.2 moles per liter of A, I required 0.1 moles per liter of B to be used. So the concentration of B went from 1 mole per liter down to 0.9 moles per liter. So here's the equilibrium constant, simply products over reactants. Now I just input all the numbers, crunch the numbers, and here's the answer right there. Okay. So this next section over here, I spent years figuring all this stuff out, and when you figure things out, you get to the point where it becomes simple. I remember taking thermodynamics courses in chemistry. It's like, okay, this is hard, but I get it. And then I was very fortunate to take biochemical thermodynamics from a guy who I still respect. He's passed away, but he actually won the Japan Prize in Biology one year. Very well-known scientist. And he was PhD in physics, actually, from Cambridge in England. So I busted my butt, and I realized that I thought I knew things from my chemistry course when I really didn't. And then I taught a 700 level course, co-taught it with him. And so we're sitting around trying to explain this stuff to graduate students, and that's when I really, wait a minute, now I understand it at another level. It's crazy. It's actually quite simple. So G is usable energy, or free energy, named after a guy named Gibbs. So Gibbs free energy. It's the amount of energy that can be used to do work. And H is enthalpy. That's the energy involved when bonds are broken and bonds are being formed. And S is entropy. That's the energy associated with changes in the state of randomness. So remember, the definition of zero entropy would be a perfect crystal at 0 degrees. So here's some examples of an increase in randomness. If I take a solid and I turn it into a liquid, the solid is, the particles can't move from point A to point B. They can vibrate, but they can't move from point A to point B, which they can in a liquid. So it's an increase in randomness. When a liquid turns into a gas, it becomes more random. When you get more molecules formed from fewer molecules, now those limited number of elements are more randomized in more different kinds of molecules. And finally, when the volume is increased. So what is the relationship of these three kinds of energy? Here's the mathematical relationship. Okay, so the usable energy is equal to the energy that comes from breaking and forming bonds, also by, or moderated by, the change in entropy, the change in randomness. And notice that the temperature is multiplied by the change in randomness because it's temperature that causes all these things to happen. So you cannot separate those two terms from one another. Oftentimes, chemical reactions, it's difficult to measure the entropy. So what they do is they ignore the entropy and they just talk about the heat of reaction, which is simply this estimation right here. Okay. So let's look at a real chemical reaction and apply these ideas to it. Here's octane burning in oxygen to give you CO2 and water and a bunch of heat. Here's the transition state in the reaction and in the energy diagram. So here's the delta G for the reactants, here's the delta G for the products, right? I gave numbers here. So it gives off this much energy, right? Now, why is delta G negative? Well, in the beginning, somebody had to make this decision. Do I take this number and subtract this number? Or do I take this number and subtract this number? Going back to the equilibrium constant, how do I define it? Do I put reactants over products or products over reactants? Somebody had to make those decisions, and they're related. So the decision was to take this number and subtract that number. So this turns out to be a negative number. That means that an exothermic reaction like this gives a delta G of a negative number. So let's see. We'll use the same chemical reaction, and I'll point out a couple of things. I can take gasoline, put it into a bucket, throw a match on it, and I can see all this heat coming off, right? But I have no way to measure the delta S, so I can't measure the complete delta G. So open systems cannot harvest energy, and this is energy, so we can only measure the delta H. So going back to what I was explaining up here, this is what I can measure in an open system. I have no way to measure the entropy. But we have designed machines, such as the internal combustion engine. In biology, we have proteins, and these are able to harvest the delta S, so we can do mechanical work, and then we can actually measure the delta G. So what does the internal combustion engine use? It uses expansion of hot gaseous products to push the piston. By pushing the piston up and down, that's work. Work is force times the distance. So the energy to do the work is defined by this. So what kind of things are causing this piston to move? Okay, let's see, where am I over here? We have a liquid turning into a gas. We have more molecules formed from fewer molecules. And the volume is increased, and that's where we harvest the mechanical energy. The volume increases and pushes the piston down. And proteins use the movement of atoms when bonds in the substrate react, are broken to harvest more energy to do mechanical work. And again, it's conformational changes. So say I have a protein in this conformation. Maybe a molecule of ATP is involved in this. I hydrolyze the ATP and I just cause this part of the protein to move relative to the other part of the protein. I just did work on this protein. That protein may be doing mechanical work of some other type. So you may have heard of ATP, and in your biology books, this is what you see. The delta G is negative 7.3 kilocalories per mole. That negative number means that the reaction will happen. It will be an exothermic reaction. That's what this means right here, right? So we took this value and subtracted that value. Okay. So here's some ways of harvesting mechanical work in biology. So you can tell that this is not me because those muscles are so small. But anyway, here we're doing mechanical work. Look what's happening to the muscle. Here the arm is extended. And then look at the muscle. The muscle has gotten fatter, right? It's stretched out and now it's bunched up. How is that happening to produce this mechanical movement? Well, notice that the muscle is also attached to the bone over here. And it's interesting how you look at different kinds of animals with different kinds of strength and different types of muscles. You can imagine if this muscle right here was attached to the bone over here, we would be able to get more leverage. This would be a stronger arm, but it might also be an arm that could be damaged more easily by trying to do too much work, by ripping off that muscle from the bone. That'd be a problem. So here's what happens. Here's the relaxed form of skeletal muscle. Here's the contracted form. So what you see is this. And then during the muscle contraction, you see this. So you shorten the distance between these lines right here, these Z lines. You make the muscle shorter by all the muscle proteins acting at once to shorten. So here's a little bit closer detail of what's happening. Here's the actin, which is the thin filament. Here's the myosin, which is the thick filament. So here's the thick filament. Here's the thin filament. Okay. So ATP is bound to it. And then ATP gets hydrolyzed. And during the hydrolysis of this ATP, you see this movement of part of the myosin molecule. That allows it to attach to the thin filament and then cause this motion to happen. So it attaches over here, pulls it that direction, thereby shortening this distance between the two Z lines. So this is a conformational change that happens upon a hydrolysis of ATP. Here's another example. The sodium-potassium ATPase in the nerve cell membrane that uses ATP to pump 3 sodiums out for every 2 potassiums that it brings in. So here's outside the nerve cell, here's inside the nerve cell. So here's an ATP molecule, okay? So here's 2 sodiums, or we got some sodiums in. So 3 sodiums go in, ATP is hydrolyzed, and look, it causes a conformational change in this membrane protein. So that allows the 3 sodiums to go, the 3 sodiums to go out. And then, since this is now in a different conformational change, it doesn't bind to the sodium anymore, but it binds to the potassium. So 2 potassiums bind here. Then upon that, it causes a conformational change. Again, it causes the release of the potassiums. So what you've done is you've taken 3 sodiums and put them out, taken 2 potassiums and put it in. So 2 things happen now that allow this membrane to be active. One is you have an electrical component. It's positive on the outside, negative on the inside. The membrane itself is an insulator. The other thing is you have a high concentration of sodium outside and a high concentration of potassium on the inside. So when a depolarization event occurs, for example, when a neurotransmitter binds to its receptor on the surface of this nerve cell, then it causes that channel to open up. So when you do that, sodium rushes in, potassium rushes out. But this happens for a fraction of a second because all biological signals are instantaneous. They don't stay open. It's like that. Okay. The lifetime of most neurotransmitters is that. If you want a prolonged signal, you have to keep producing, because every single one lasts only like that. So then I just thought this was a good diagram that shows you a nerve cell, all the different components. So here's the endoplasmic reticulum, here's the nerve synapse over here, and you can see that it's interconnected to all kinds of other nerve cells. It forms a network. This myelin sheath right here is when you make a long connection to another nerve fiber. And myelin is a protein that essentially acts like an insulator for a wire. Anyway, so that's it. We made it to the end of the course, and yeah. So there's me with my bad jokes. There's you. You just can't stand it anymore. But yeah, you made it to the end. So somewhere in the future will be your pot of gold. So that is the last lecture for the semester. And, here's my schedule. We're going to have a review on May 11th for Unit 5. And you got a couple of days before you have to take your final. Because all dates and everything else are kind of thrown out because of the virus, I just made the last 2 days of final exam period your final exam. So you have homework 21 due tonight, and then for the next several days you have homework due every night. So my plan will be we'll upload all the answer keys on May 11th. May 14th, exam 5 will be open, same on May 15th. That gives me a couple days to finish grading and do grades. So anybody have any questions about anything? Viruses, questions about the meaning of life, religion, food. So if you don't have any questions, it doesn't mean you can't answer or ask questions by email or in the discussions. And let's see. So John is saying there's a few issues with homework 21. Well, I don't know what they are, so. Okay, I'll check that out, John. It sure is a lot of work trying to get everything to go online. So if you don't have any questions, I'll just close the lecture, and then within about an hour, hour and a half, it should be available up on YouTube. And if you got any questions, just email me and use discussions and private messages, and I'll look in there too. Okay?
Yes.
Well, thank you. See you.
Bye.
Bye.